Hi everyone, happy Friday. Welcome to the Myotonic Dystrophy Foundation's Meet the DM Drug Developer session. I'm Tanya Stevenson, the CEO of the foundation, and today we're going to be talking with the PepGen team. Just a little bit about the foundation. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatment and a cure. A reminder of some of the resources and support that are available from the foundation. Our toolkits and publications are available at myotonic.org/toolkits-publications, and that includes our clinical care guidelines that our community can use to share with their clinical care teams. Our support programs can be found at myotonic.org/find-support. Our calendar of events, including our support groups, can be found at myotonic.org/calendar/month. And our digital academy, which includes all of our past presentations and webinars, including sessions from our conferences, at myotonic.org/digital-academy. Today's program is being recorded, so you will also see that on the Digital Academy over the course of the next week or so. So today is our second of our series for 2023 for our Meet the Drug Developer series, and we have several more for the rest of the year up until our conference in September. Most of these are happening on the first Friday of the month, but we do have a couple of changes. So next Tuesday, we will have a presentation from Avidity Biosciences, which is looking at special top-line data. And then in June, instead of the first Friday of the month, Artex Biotech will be presenting on the second Friday. In July, we will be talking with AMO Pharma, and then in August again with Avidity Biosciences. We hope to see everyone at our conference in Washington, D.C., September 7th through the 9th. Just as a note, September 7th will be MDF Advocacy Day on Capitol Hill. So we will be meeting with senators and representatives to make sure that they're aware of myotonic dystrophy and that they are pushing for more research for myotonic dystrophy as well. You can go to myotonic.org/conference to learn more about that. Okay, it's time to meet the four members of the PepGen team that are here with us today. Dr. Jane Larkindale is the Vice President of Clinical Science at PepGen. She's dedicated the past 15 years of her career to accelerating therapy development for neuromuscular diseases. She launched and ran international consortia and programs focused on data standardization and aggregation and use of the data to support regulatory acceptance of disease models, outcome assessments, and biomarkers to accelerate drug development. She has worked with world leaders in the rare disease space, including the Critical Path Institute, the Muscular Dystrophy Association, and the Myotonic Dystrophy Foundation. Also here with us today is Stephen Babcock, the Senior Clinical Project Manager at PepGen. Stephen is a clinical research professional with 15 years of industry experience in a variety of different therapeutic areas, specializing in the conduct of phase 1 through 4 clinical trials. He holds a Bachelor of Science degree in biomedical engineering and a Master's of Science in clinical and regulatory project management. Joining the team today, we have Ashling Holland. She's the Director of Preclinical Development at PepGen. Her international scientific research career over the past 12 years has focused on multiple types of muscular dystrophy. She's been leading the preclinical development of PepGen's myotonic dystrophy and Duchenne muscular dystrophy programs for the past 5 years. In 2020, she joined the board of directors of Muscular Dystrophy Ireland and is also the chair of their research committee. And Jenny Shoskes, Associate Director of Clinical Development. She's a pharmacist by training and has worked for the last 8 years within the rare disease drug development. space. Prior to joining PepGen, she worked within clinical development at Fulcrum Therapeutics and Apellis Pharmaceuticals, where she supported neuromuscular programs, including FSHD and ALS. Thank you to everyone who submitted their questions in advance. We have provided those to the PepGen team, and they've done their best to integrate them into the presentations that you will see shortly. If your question wasn't answered or you have additional questions, you can ask those live. Just open the questions tab, type your questions, and click send. It will get to our team here. If you're on a smartphone, click the question mark icon at the top of the screen. You can type your question and click send. If today's program speaks to you, please consider showing your appreciation with a donation to MDF. You can do that at myotonic.org/donate. Okay, without further ado, I would like to welcome the PepGen team. Thank you everyone for joining us today.
Hi, and thank you very much for that great introduction, and thanks to the MDF team for giving us the opportunity to tell you more about what we're doing at PepGen. We're really excited about what we're doing for myotonic dystrophy type 1, and we really hope that we can get you excited about it also. So our lead candidate is called PGN EDODM1, which is an enhanced delivery oligonucleotide designed to address the central cause of myotonic dystrophy type 1. That's a whole lot of words, and I'm going to explain a lot more about what that means as we go in. If I can change my slides, there we go. Disclaimers, you can read those elsewhere. That's just the information we always have to share with you. So what we're going to be talking about today, and I'm going to be passing the baton between my colleagues, is I'll give you an introduction to the company and our technology, and then I'm going to pass it over to Ashling, who's going to talk about why we think that EDODM1 may make a difference for people with myotonic dystrophy type 1. And as Tanya said in the introduction, Ashling's been working on this project for many years, and there is nobody better to tell you about how it works and what data we have. After that, we'll hand over to Steve, who'll give you a bit of background about clinical trials and where we are. And then Jenny, who will talk about what we've been thinking about as we're moving towards our first clinical trial in people with myotonic dystrophy type 1. And then coming to the end, we'll come back to me where I'll talk about how you can be involved and the next steps for us. And we hope to leave plenty of time for question and answers at the end, because we've got some really good questions ahead of the webinar, and I'm sure you'll have more by the time we get to the end of this presentation. So PepGen is a small company. We're headquartered in Boston. You can see us all at our summer picnic last year on that top left panel. We are a rare disease clinical stage company. We really have two lead programs. One is a myotonic dystrophy type 1, the other is in Duchenne muscular dystrophy. But our technology has the potential to address a number of other diseases which are coming up in the pipeline before. So we're really excited to talk to you mostly about our myotonic dystrophy type 1 program, but this technology has applications to other diseases as well. This is just to show you who we are, the people who are on the phone right now, and just sort of show you how we feel about our company. You are represented in the middle of this graphic as the myotonic dystrophy community, because you're the people we're working for. Everything we do centers around what we can do for you and how we can move our potential treatments forward. You can see we have a number of different functions within the organization. This is just a subset. I didn't include legal and finance and many of those functions. These are the ones who really directly contribute to developing a drug. And you'll hear from Ashling, who talks about preclinical research and development, Jenny, who's going to talk about the clinical development as we move towards clinical trials. I'm going to talk about how we work with the community, and Steve is going to talk about operations. I did want to point out Elena, who's our head of patient advocacy, is currently out on leave, so she's not here today, but know her face. She will be at the MDF meeting. She'll be around, and I hope many of you will have the opportunity to work with her in the future. So with all of that preliminary, what is the EDO technology? EDO, I mentioned earlier, stands for Enhanced Delivery Oligonucleotide. Let's break that down. So you can see here under the box that says the challenge. There are these drugs or molecules that are called oligonucleotides, which have been being tested in myotonic dystrophy for a really long time. I remember way back when I worked for the Muscular Dystrophy Association talking to Charles Thornton and Thurman Wheeler about these oligonucleotides that could potentially be used to treat myotonic dystrophy. But for years and years, including the first clinical trial that was done in people with myotonic dystrophy, there was a fundamental challenge with these oligonucleotides, is they work really well if they got into the nucleus over here, but they didn't. They got stuck outside the cell and they couldn't get in. And we sort of, as an analogy, like to say it's like you're the person with the most important piece of information for a call, but you can't connect to Zoom, so you can't share that information and nothing can be done. Well, now something can be done. This is what our EDOs do. That's represented by this little blue peptide on the end of the oligo. These are carefully designed peptides that were developed at the University of Oxford and the University of Cambridge over a period of 10 or 15 years. And what they do is they pick up that oligonucleotide and let it into the Zoom room where that critical information can be shared. And we hope that this oligonucleotide can do what it's designed to do and hopefully treat myotonic dystrophy. So I'm going to take you through a cartoon. I think most of you know that myotonic dystrophy is caused by an expanded CUG repeat in a gene called DMPK. So this is in unaffected individuals. We have a short repeat sequence in the nucleus that allows a protein called muscleblind to go out into the rest of the cell and go about its usual business, which is making sure proteins are processed normally. In people with myotonic dystrophy, the CUG repeat is expanded, and essentially it acts like a sponge, and it sponges up all their muscleblind so it can't get out into the cytoplasm, out into the rest of the cell. It can't do its job, and you end up with a whole lot of misprocessed proteins. Those proteins all have a function, and they're supposed to do something, and that results in the many, many different symptoms of myotonic dystrophy. What happens with our drug? So there's PGN EDODM1, our molecule represented by a green dotted line. It comes into the cell, that's what our peptide does, and it binds to those CUG repeats. And that essentially means that the repeats no longer can act like a sponge. The muscleblind can get out of the cell as it did in the unaffected individual and do what it's supposed to do. Proteins are processed normally, and we hope that will resolve the symptoms of myotonic dystrophy. And Ashling will show you a whole lot of the data that we have to suggest that this is the case, that we can indeed correct those proteins, release the muscleblind, and even in mice, correct some symptoms. We recognize that myotonic dystrophy is a multisystemic disease. It affects all kinds of parts of the body. Although it's called myotonic and we think of myotonia and skeletal muscle as the main target, we know it affects the heart. We know it affects the gut. We know it affects breathing, and we know it affects the brain. Our EDOs have been shown to get into all of those systems. So, and given the mechanism of how myotonic dystrophy works is the same in all those systems, if we can get the drug into any one of those systems, in theory, we should be able to affect multiple different symptoms of disease. And that's what we're going to be just finding out soon. But first, let's take you back in the journey, and I'm going to hand over to Ashling to talk about what we found out and learned so far in preclinical models.
Thanks, Jane. Hi, everybody. My name is Ashling Holland, and as Tanya introduced me already, I'm the Director of Preclinical Development at PepGen. So as Jane mentioned, we are in the preclinical stages for our EDO-DM1 compound and hoping to go into clinical trials soon. However, I'm going to give you a snapshot today of some of our preclinical research, both in cells and in the mouse model as well, of how we believe that our compound is working and could have a positive impact for those living with DM1. So first, we've evaluated our EDO-DM1 compound in vitro in muscle cells from an individual with DM1. So as James showed in the cartoon earlier, we have one of the molecular hallmarks of myotonic dystrophy type 1 is that foci. It's these sponges that we can see that absorbs all of the MBNL1. And on the left-hand side here in the image with the blue with the red dots, these are those foci. These are the kind of the pathogenic part of DM1 where MBNL1 acts as a sponge to and is bound to. On the right-hand side here with the green image, we can see with the white dots where we can see these little quite green sharp images, and that's the MBNL1 protein binding to the foci. And again, In DM1, when MBNL1 is bound, it means that there's a global missplicing situation that's happening. But when we treat these cells in vitro, even for just 24 hours, we can see that we're reducing the number of foci per nucleus. So on the bottom left-hand image, we can see that there's far fewer of these red foci in the cells. And when we quantify this as well, we can see that at the top dose concentration, we've got up to a 54% reduction in the number of foci. But importantly as well, we're able to see that we can liberate, or we can free MBNL1 from these foci as well, allowing it, as Jane showed in the cartoon, to return back to its normal location so it can carry out its normal function and allow for proteins to be processed in the correct way. On the next slide, we've been able to take this another step forward. So we've been able to show that we can reduce the number of foci, we can free MBNL1 from the sponge, but also we can see that when we look at the downstream impact of that it's actually having an activity as well. So one of the ways that we evaluate this is by looking at missplice correction. And for this, we look at a myriad of different transcripts and we evaluate all of those in parallel together. And we can see that in DM1 patient cells, again, we're looking at a muscle cell line in particular here, we can see a dose response when we treat with EDO-DM1 and correcting this missplicing profile. And at the top concentration that we evaluated, we saw almost 70% missplicing correction. So again, combining the initial data that we have for reduction of foci, release of MBNL1, and now correction of downstream splicing. Together, all of this from the in vitro perspective is showing good promise for translation into the clinical side of things as well. However, we know that with our mechanism, we're doing a steric blocking approach. We're not trying to degrade the amount of DMPK there. And again, as Jane mentioned, DMPK is where that repeat expansion is, and we're not trying to break it down. However, when we looked at DM1 patient cells, again, looking at the muscle cells, even at the top concentration that we evaluated, where we could see correction, we didn't see any changes in DMPK levels, which is very important for a potential safety side of things as well. Also, we took this another step forward and we looked at it in monkeys as well, or non-human primates. Again, for this, we looked at it from a repeat dose perspective, so not just a single administration. So for this, we looked at 3 administrations which were given every 2 weeks. And even at the top concentration evaluated as well, we didn't see any degradation or breakdown in the DMPK levels. So again, this is further supporting our mechanism of action that we are not intending to break down DMPK and is supported across multiple preclinical models as well. On the next slide, we've taken one of the most routinely used and well-established mouse models in the field for myotonic dystrophy type 1. This is the HSA-LR mouse model. And this mouse model is incredibly powerful for preclinical evaluation and preclinical research. So just as a bit of a background into this mouse model, it's got about 220 to 250 of these CUG repeats that are present in the human skeletal actin transgene. And again, just to make it a little bit less sciencey as well, we know that this mouse model, it only has a skeletal muscle phenotype. So it doesn't have the full same kind of manifestation as what you would have in humans in this model. However, it does have some of the key pieces of it. So there's foci that are present there. The MBNL1 is impacted in the pathology as well. We've got the ability to evaluate missplicing profiles, but also we can evaluate myotonia in this model as well. So our first look into this was looking at the missplicing profile for the HSA mouse. And we looked at this after a single dose. And we could see correction. And here we're just showing 2 of the transcripts that we looked at, even though we looked at more. But again, just for example, we've been able to show here that we can correct for ATP2A1 up to 91% correction of this transcript, and for CLCN1 up to 68% correction. And both of these transcripts are incredibly important for the muscle excitation side of things. However, again, similar to how we were able to show correction in the muscle cells in vitro, we can show great levels of correction as well in vivo in a mouse model. However, we can also look at the functional level, as I said, in this model. So we can look at the myotonia profile. And again, after a single dose at 30 mg per kg, we were able to see complete correction of myotonia, and we can restore it to what would be a normal wild type level that we can see in control mice. So combined, this data is really supportive from the translation from the in vitro to the in vivo side of things. And again, we were able to look at this even more detailed way. So after a single dose on the previous slide, we were able to show correction 2 weeks post-dose. But here we've looked at it both 12 weeks and 24 weeks post-dose. And we can see using these same two transcripts as an example in both the gastrocnemius and the quadricep muscles that we've got sustained levels of correction out to 24 weeks. And again, this is just after a single dose treatment showing highly durable levels of correction in this mouse model. And this is incredibly important to help us when we start to think about the clinical design for our program as well. And showing that we're able to have correction out to 24 weeks is very promising to show and to potentially suggest that we might not need to dose too frequently in the clinic either. And on the next slide, we have just a little bit of a highlight here from some of the data that I've just presented, in addition to other data that we weren't able to present today just because of the timings. Again, with our whole preclinical package that we've developed so far as the team at PepGen, we've been able to show very promising data. So we were able to show reduction of the toxic foci and liberation from MBNL1 in the DM1 cells. We've also been able to show misplice correction in 2 different types of preclinical models, one with long repeats and one with short repeats. Within our DM1 mouse model, we've also not just been able to show this robust misplicing correction, but importantly, the reversal of myotonia after a single dose. And again, as I mentioned already, we were able to see durable, long-lasting correction out to at least 24 weeks post-single dose. And as we've kind of shown already today, what our mechanism is for freeing MBNL1 and not degrading or breaking down DMPK. As I say, we've been able to show that we do not break that down either. And we haven't really gone into any of the safety or the tolerability studies that we do in animal models. However, just to give a slight insight into what that's been showing so far, we've been able to see that up to 90 mg per kg, so high dose levels in non-human primates, we were able to see that it was well tolerated after a single dose. And what's important for our program, for our platform more generally as well, is that with our EDO technology, we've been able to show pharmacologically active levels of oligonucleotides to muscle in humans with one of our other programs, but also from the technology more broadly as well, we've been able to show delivery of oligonucleotides to the central nervous system, so into the brain in non-human primates, which could be very valuable for the therapeutic side for myotonic dystrophy type 1. So that's just a quick tour and a snapshot into our preclinical development, and we're really excited about the promise of our technology so far. But I'll hand it over to Steve and he can talk us through the clinical trials and where we're going to be bringing this data to next.
Thank you very much, Ashling. I'm Steve Babcock, and I will be talking to you about what a clinical trial is and how we select and start up our sites. So clinical trials are part of the drug life cycle. So it starts with research and preclinical development. All the information and tests that Ashling has just brought you through falls into this bucket. Then it progresses into human testing. So this is the clinical trials phase. After that, all of the data from the trials is compiled into a submission to a regulatory authority for a specific country to see if we can get approved for marketing and selling that drug within the country. If the drug is approved and it becomes available, there's also a surveillance period that occurs afterwards where additional safety and additional efficacy information is still reported on the drug. Currently, the Freedom DM1 trial is getting ready to begin, and clinical trials are conducted in phases. In general, the later phase corresponds to an increased number of participants and an increased number of sites. So phase 1, 2, and 3 are prior to the review and approval by a regulatory authority, and phase 4 occurs afterwards during the surveillance period. So to focus on phase 1 studies, they are to evaluate the safety. What we're looking for is to find a safe and tolerable dose. And this usually starts first in humans with one dose of the investigational drug. And we look to test in healthy volunteers or affected populations. And we also look at how the drug interacts with the human body. The Freedom DM1 trial is a first-in-human phase 1 trial, up and coming, as Ashling mentioned. The clinical trial process can be broken down into 3 stages. First stage, startup, second stage, maintenance, and the third stage, closeout. So in startup, that's where we do all of our study planning. We select our vendors, we get them set up. We submit to the regulatory authorities for the ability to test on an unapproved medication, and also the ethics committees to make sure that the protocol that has been developed is ethical and isn't exposing anyone to any type of unnecessary risk. Also, the site selection and site startup process begins and occurs during this phase of the trial. And this is currently what we are getting into with the Freedom DM1 trial. The maintenance phase is once the trial is open for enrollment, there is active monitoring of all of the data coming in. Adjustments are made as needed to the study in order to make sure we are getting the highest quality data and making sure participants stay safe. Then after the enrollment is completed, the study is in the closeout phase, which is where we do all of the final data cleaning and preparation and analyses for our regulatory submissions. And afterward, the results may be published as well. So one question we've heard from the community is, where are the sites going to be for your trial? Why is there not one in my area? And how do you decide where the sites are going to be? So I'd like to shed a little understanding on how we select our sites and what the site selection and startup process looks like to help answer that question. So it begins with feasibility, which is when we start looking at the different countries where we'd like to try to test our product. And we start reaching out to investigators, we give them questionnaires, we gather as much information as we can from investigators within those countries that are interested in participating in the trial. So that leads to a large amount of information and a large number of investigators in the potential pool. Next, we start our qualification process. So this is where we investigate and learn about the site's experience, the site staff and equipment and capabilities of the site, and also how their patient population looks and if they would be able to participate in the trial that we've designed. So from that pool of investigators from which we've identified and qualified, then we do selection. So this process is very thorough, and there's a lot of factors that go into it, including the geographic diversity of where these sites are, the site startup timelines to make sure that we are going to be able to complete the trial and get the product submitted and approved as fast as possible so that patients can access these life-changing treatments. And also the results of the qualification process weigh heavily into the selection process. After a site is selected, then we move on to activating that site. So activation means approval to enroll and start the study. Also involved is recruitment activities to reach out to the community and get the community excited, informed about the trial, and give them the opportunity to participate. And also it's setting up supporting mechanisms so that the site is able to adequately support the specific patient population that is participating in the trial. And what I'd really like to make clear here is that even if there is not a site in the immediate geographical area, there are travel provisions or ways that you can, or a participant can join the study without having a site in close proximity. So that includes airfare, hotels, travel arrangements being made, and Basically, they will help anyone who is willing to and able to travel to participate in the trial to be able to do so. And with that, I will pass it off to Jenny.
Thanks, Steve. Hi, everyone. As you mentioned, my name is Jenny Shoskes, and I'm an Associate Director of Clinical Development here at PepGen. So Steve just kind of went through what goes into getting a study up and running and how we make some of the decisions around the trial and which sites. What I'll be focusing on is how we actually design our trials, what we consider, and why we make certain decisions. So you start off by thinking about the purpose and the phase of your trial. As everyone went through, we just finished our preclinical development, and now this would be the first time looking in participants or people with DM1. So the primary objective is safety. Then we think about our patient population specific to the trial, and really there are two things that we consider. Number one, again, is safety. And the second is the ability to have the fastest path forward. So we need to make sure that we're selecting a group of patients that we don't put at an unnecessary safety risk. So maybe that they don't have any significant medical problems to begin with. So this is why you might see age restrictions or restrictions around certain conditions. We also want to see if we can do something called enriching a trial. So that's selecting for a group of patients in a trial that we think might be able to show change in a shorter period of time. But that would still be representative of the full population. So sometimes if you see inclusion or exclusion criteria, specifically typically around function or how the patient is presenting at that one point, it's not necessarily because the drug wouldn't work in that population or people that don't fall into the trial. It's just because we're trying to do things in a safe way and in a way that might be a little bit faster. Importantly, we think about the drug itself. So in our case, EDO-DM1. For instance, our drug is given via an IV infusion. And Ashling showed some data about how long we see that effect for. So that's important when we think about how often we dose the drug and when we actually measure things in the trial. Ultimately, once we know that the drug is safe, on top of that, we want to see if the drug is having an impact in meaningful ways. We do this by selecting and measuring different clinical trial endpoints, but we compare it to something called a placebo group. That's a group of patients that are not on the drug, and that's a way to compare to see if it's really the effect of the drug or if it's just the effect of being on a trial. Really, we go and select endpoints that we want to be clinically relevant to you, the patient, to caregivers, to clinicians, and also to regulators. And we want these endpoints not just to be relevant, but to be susceptible to change over a defined period of time. So we might know that something is important to you or your disease, but it might only change over a period of 2 years, making it not the best choice for a clinical trial endpoint, but still important overall. On the next slide, we go through a couple of types of clinical endpoints and examples of them. I know one of the #1 questions is, what is the design of your trial? How many visits are there? What things are you looking at? And at this point, we can't share that as we're still working with regulators, but I'll go through kind of buckets of what we typically look at and why we do so. So the #1 thing is safety. So we measure different things like blood or urine or your vital signs like your heart rate to make sure that the medication itself isn't having an impact on those safety measurements. Then we look at pharmacokinetics or pharmacodynamics or PK and PD. These are long words of saying what the body is doing to the drug and what the drug is doing to the body. So those two things really are looking at the concentration of the drug in the body in the different areas, as well as how the drug is interacting once it's in the body. So in DM1, we look at changes in splicing to see if we're really able to liberate that muscle blind or that MBNL1 and have those downstream impacts to those different proteins that we were able to show in that mouse model and those cell lines before. Next, and importantly, we think about how the patient is functioning. So we want to see if the drug is able to have an impact on your disease. So we look at this in a variety of different ways, looking at symptoms such as myotonia, strength, your ability or the time for you to do certain tasks that are impacted by the disease, and looking at your organs, your body systems that might be impacted by the disease. Now, most importantly, we might be able to see a 0.025 second change in one of these measurements, but at the end of the day, we want to make sure that the changes that are seen are important to you. So we do this by asking and using patient-reported outcomes. They're typically surveys, and they ask patients how they feel or function. As we've thought about our clinical trial study and design, we have engaged with the DM1 community and have been able to incorporate your feedback to ensure that the endpoints we include are meaningful to you. So with all that information has led us to our Freedom DM1 trial. It is a phase 1 study, as it's the first time we're going into people with DM1. And it is planned to be conducted in around 24 people with DM1. It's called a single ascending dose study. So essentially, as Steve mentioned, typically in the beginning, you start off with one dose of the drug and you check the safety to make sure everything is okay before you ascend or move up to the next dose of the drug. It will be a randomized, double-blind, placebo-controlled drug, so there are trials, so there will be a placebo group so that we can compare to. And some of the main things we'll be measuring are safety, correction of splicing, as well as some of those functional assessments. We are planning to open this trial in the first half of this year, which, as you probably will notice, is very soon. And we hope to have data on this study within the next year. So I'll pass it to Jane, and she'll also be able to tell you a little bit more about how to find out more when we are able to release more about this trial.
Probably works better if I go off mute. Thanks, Jenny. That was a great introduction to the Freedom DM1 trial, which we're really excited will be coming soon. We really hoped that we'd be able to tell you more on this webinar about the details, but as Jenny said, until we've got full alignment with the regulators, we can't tell you much more, but I will explain how you can get that information as soon as I'm allowed to share more details. So what I'm going to talk about in this final part of the webinar is how we operate within the community. We've already referred to the fact that everything we do is guided by you, the DM1 community. We really like to hear from you. What we're doing is for you, and we want to make sure that our drugs work for you and our trials as well. So everything we do requires community insight and input. People always ask, I put up the slides, what about preclinical targets? What do you mean by that? Well, we've heard from you, the community, many times is, what about DM2? Are you developing anything for DM2? Well, unfortunately, EDO-DM1 is specific for the repeat sequence for myotonic dystrophy type 1, but there's no reason that we can come up with why a similar approach wouldn't work for DM2, and that's something we're very interested in exploring. So all the way back to the preclinical work, we're listening to what you have to say and try to work for you, the community. In terms of things like therapeutic preferences, and Jenny referred to this, is we don't want to measure something in a study or correct something in a study that doesn't matter to you. If you as a person with myotonic dystrophy and I go in with a drug and say, it's great, I can cure the pain in your left little toe, you probably don't care very much because you've got an awful lot of other symptoms that are much more significant than the pain in your little toe on your left foot. So we really like to hear from you as to what is important for us to correct to see if our drug does, and to then select the right endpoints so we can measure things that are important to you as well. And that all goes to informing our clinical trial protocol to make sure we're measuring things that are important and developing a trial which you want to take part in. If I told you you had to stay in the clinic for the next 17 months, you'd probably say no. You've got jobs, you've got lives, you've got families, and that's not a feasible thing to ask of anyone. So all of those things are the sorts of things that we need your input on. And we also ask for input on the materials we build to tell you about what we're doing. We get feedback in a whole lot of different ways. I'm going to show you some of these as we move through the next slides. We have in place a standing community advisory committee, which is one of the most valuable groups we have. This is people with myotonic dystrophy and their caregivers. We bring them together at regular intervals. We've had 3 advisory committee meetings recently to really talk through some of these things that we need to know so that we can develop drugs that are meaningful to you. What are the symptoms that matter to you? Here's our trial design. What do you think? Are we making people come to the clinic too often? Are we making people stay too long? Are we taking your blood too often? What do you need in the clinic to help you understand what's going on? What accommodations do we need for travel? Steve referenced this. It's very important to us that even if we don't have a clinic in your area, if you want to participate in the trial, we want to make it possible for you to travel to the site. But we know travel's not easy. Some of you may not be able to travel by yourself. You might need a caregiver to come too. So we've made sure that our concierge services allow for that. Some of you use wheelchairs. We need to make sure that any accommodations make it easy for people in wheelchairs to stay there. So we're really thinking through what we can do to make this trial easy for people with myotonic dystrophy to take part in. And we also really work with our advisory committees on the best way of sharing information with you and getting information back. So this is a lovely example photo of my colleague Elena and I talking to one of our community advisors at the MDF meeting last year. We like to attend meetings and we like to be there, not just to tell you about what we're doing, but also to hear from you. And as a good example of this, I had 3 or 4 different people at the conference last year. When I asked them what's important to people in terms of symptoms, they said swallowing. So I brought that back to our clinical team and we went off and researched every way that we could measure things that were measure swallowing in people with myotonic dystrophy, because we know that our drug gets into smooth muscle. It should affect swallowing, but we've never really been able to measure it. So we went back, we figured out what we could measure and incorporated that into our trial, because people told us they care about that. And that's how we like to operate. In terms of communications, we have many channels by which we communicate with you, the community. And some of these are written materials. We have a video that's coming soon. We have meetings like this. A lot of our written materials we like to run by some of our community advisors to get their feedback. And an example of that is the one-pager you can see on the right hand of the screen, which we developed, I wrote, and I thought it was great. I shared it with a couple of our community advisors and they just handed it back to me and said, too scientific. You go through a page and a half before you get to any information I care about. You need to rearrange the order, simplify it, and I suggest this order. So we went back and we rewrote it. We reordered it so that hopefully it is more useful to you, the community. And we do that with everything. One of the pieces of feedback we got from one of our advisory boards was that it would be very helpful for participants in a trial to have a planner that told them what was going to happen in the trial on each day, each visit, what they needed to bring, how long it was going to take, and basically lead them through every step of the way. Again, we developed that planner. We've had some of our advisors read through it and give us feedback on it, and that will be ready to go as the Freedom DM1 trial starts. So to the question you're all asking for. I know this is the question everyone says, is how can we learn more? When's this trial opening? What can I be included? Where is it going to be? I can't share that information now, but very soon we will be able to share that information. And here's just a list of places you can go to learn more once we are open. Clinicaltrials.gov is a government website that lists clinical trials. MDF is always a fantastic resource. I always recommend talking to MDF, and we will share materials as we can with them to share with you, the community. Always recommend talking to your doctor. Your doctor is going to be a great resource for you. But also follow us. Our website's pepgen.com. We are on Twitter and LinkedIn. And if you ever have any questions, you can just email us at community@pepgen.com. And with that, I'm going to stop talking. I'm going to go off the slides in a moment so you can see us and answer some questions. But I want to finish with a huge thank you to you, the community, our advisors, and the Myotonic Dystrophy Foundation for inviting us to speak today, and all the other wonderful work the MDF does. we are really excited to be where we are, beginning to move towards our first clinical trial. We are confident in our technology. We really hope that the trial is informative and that we will be able to share with you data next year as planned. With that, I'm going to stop sharing my screen and come back and invite my colleagues back on so we can answer some questions. Okay, so please feel free to put your questions into the chat if you come up with them as we go forward. The first question we got from multiple people is the question we've always heard many times, which is about whether we have the potential to treat myotonic dystrophy type 2. And I already alluded to this earlier, but Ashling, do you want to expand on that a little bit?
Sure thing, happy to. So the data that we presented today is all around and focusing on myotonic dystrophy type 1. Again, though, there's a similar approach that could be applied for myotonic dystrophy type 2, and we would need separate preclinical development and clinical development for that. And applying our EDO technology and our platform to DM2 is something that we think about a lot as well.
Thanks, Ashling. The next one is a similar related question, which is, can EDO-DM1 be used in people under the age of 18? And the answer to that is we don't know. We haven't tested it in children. Obviously, we haven't tested it in any humans. But theoretically, based on the mechanism, there's no reason this wouldn't work in congenital muscular dystrophy in a similar manner to how it works in adult muscular dystrophy. But because of the concerns around testing in children, our first trial will be in adults. But there's no theoretical reason why it couldn't be used for congenital. Okay, the next question is, what symptoms do we think that EDO-DM1 could affect? We've referred to the fact that we can get into the CNS, but what about the heart and the lungs, and does it build muscle? Ashling, how about you address that one as well?
Sure thing. So another great question. So with our EDO platform, we've been able to see that some drug can penetrate into the CNS tissue. However, there's more research for us that needs to be done on that side of things. You mentioned then about what about the heart and lungs as well. So thinking about it from our EDO-DM1 perspective, and just with the call that we have today, for the data that we have at the moment, in the mouse model that we've used, it doesn't have the cardiac phenotype in it. So we weren't able to evaluate it in any of the work for myotonic dystrophy type 1 at the moment. However, with our platform, we've been able to show that in non-human primates, we can get drug into the heart and into other critically impacted tissues as well. So we believe that when we translate to the clinic, there's the potential that we could see a benefit there as well.
Thanks, Ashling. Another question that we've had several times before, and is always a good question, is, will EDO-DM1 slow progression or improve the disease? Eventually, we get the very question, will this cure myotonic dystrophy type 1? The answer is, again, we don't know. Our animal model is imperfect. We don't know. Certainly, in the animal model, as Ashling showed you, we showed improvements in myotonia. Our suspicion is that there'll be some symptoms you can correct, some symptoms you can halt progression. We don't know, and this is something we'll be evaluating in detail as we go into clinical trials. Next question is, how long will effects last? And I think related to that was the question of how frequently we're going to have to dose. And again, I seem to be directing a lot of questions to Ashling, but Ashling, this is probably for you.
Yeah, sure thing. So again, at the moment, we're working off a lot of preclinical development data that we have. So again, looking at the mouse model, as we've kind of mentioned already, we've been able to show great durations of activity in the correction of the splicing profile. So as we said, we've got, after a single dose, we can see misspliced correction out to 24 weeks. Again, with the clinical side of things, we anticipate that we'll need repeat dosing. And as Jenny has already suggested from our phase 1 clinical trial, we're hoping to be able to get a better understanding for the frequency and dosing that we'll need as we progress through the clinical development stages. So more to come on this.
Thank you. And as you were talking, a follow-up question came in. As you said, it reaches the CNS and gets into the brain. What does that mean? Again, Ashling, that's for you.
Yeah, sure thing. So we have data from our EDO platform, and we've been able to show with our EDO 51 program for Duchenne muscular dystrophy for individuals that are amenable to exon 51 skipping, we were able to see that in non-human primates that at higher dose levels, we were able to penetrate into parts of the CNS, so able to get some concentration both into the cerebral cortex and into the cerebellum as well.
Great. And unfortunately, we don't know exactly what that means in terms of symptoms we may be able to correct, but at least we know we get there.
Exactly.
Okay, changing tack slightly to talk a little bit more about the trial. The next question we have is timing. When's the trial going to start and when will the drug be approved? And Jenny, why don't you answer that one?
Yeah, sure. So as we mentioned, are currently planning to start our first clinical study for DM1 this year, and we'll hopefully have some data the following year. As we touched on, this is a phase 1 study, so it needs to be followed by additional clinical trials. Drug development can be a slow, but it's a deliberate process in order to assess the safety and tolerability first after single low doses and then working our way up to higher doses and then multiple doses while also assessing efficacy. So we are trying to test things as quickly as possible, but also as safely as possible. And it is still an investigational drug, so we need to balance rapidly moving our program forward while still managing the risks.
Thank you, Jenny. And the next question, which I have in at least 2 places, is Steve, can you tell us where we're planning on doing this trial? You talked a bit about how we choose sites. Can you talk about where those sites will be?
Yeah, so we have completed the feasibility portion for a good number of sites, and we were reaching out in US and Canada for that, but I must caveat that with we, as Jenny said, are still in conversations with regulators. So this does not necessarily mean that those countries will be involved, but they definitely were involved in the feasibility process.
Thanks, Steve. And of course, as we move to the later phases of trials, we will need to be able to find more participants and more sites. So we anticipate additional countries being involved as we continue to develop the program. And then the final question that I have so far, which I think I'll address this myself, which is asking, are we in the FDA fast track and other programs to accelerate drug development? And the answer is not yet, but we will certainly be applying for any regulatory pathway which will help us bring these drugs forward as fast as we can. We recognize the urgency of these diseases. We want to get you effective drugs as fast as possible. To Jenny's point earlier, it has to be done in a deliberate way. We have to prove safety. We have to demonstrate we are as effective as we hope we're going to be. But we are pulling all stops to do this as efficiently as we can without taking any risks. So we're really excited to be where we are on the cusp of our first clinical trial. We hope the next time we speak to the community, we'll be able to tell you more about the trial. We'll certainly, long before that, be sharing information about this trial. And with that, I don't think there are any more questions. So with a final thank you to you, the community, and to the Myotonic Dystrophy Foundation, thank you very much for your time today. And please do contact us at community@pepgen.com if you have any further questions. Thank you.
Thank you so much, all of you. This was fantastic. We appreciate the PepGen team sharing your progress with the community today. We also appreciate the details about the clinical trial phases, your site selection process. It's really exciting to hear what the possibilities might be with this particular treatment. So I want to also say thank you for listening to our community in the design of the trial and what you're measuring to make sure it's meaningful to the myotonic dystrophy community. We are grateful for that consideration. And with that, I want to thank you again. We hope to see you in Washington, D.C. in September. Thank you, PepGen, for sponsoring our conference as well. We're grateful for that. And I'll wish everyone a fantastic Friday and a happy weekend. Take care, everyone.
Thank you.